Hey everyone, we need to talk about Unit 1. I mean, it's just so dry. I think if we're being honest, most of us would say that Unit 1 is the unit we feel like we just have to power through, that we're just trying to get it over with. Well, what if I told you that we could give Unit 1 a makeover so that it was more fun, more engaging, and more meaningful? Stick around and I'll explain how we can do all of these things using storylines in AP Biology.
Hi everyone, I'm Noel Pollard. This is my presentation, How Do Rodents Survive in the Desert, using phenomena-based 3D learning to drive student sense-making in AP Biology. I recently presented this workshop at the NSTA Spring Conference in Philadelphia, and I'm very excited to share it here with you on YouTube. Let's go ahead and get right into it. Now, if I was going to be honest, I would have actually titled my workshop, Let's Teach Science Like Mark Rober. Mark's YouTube channel has over 60 million subscribers and has amassed over 10 billion, I think, views. So Mark has really built a following, and it's all built around science and STEM. Now, who is this guy? What's his background? Is he actually a legit scientist? And the answer is yes. Mark was an engineer at JPL and worked on components that were included on the Curiosity rover. So Mark Rober, Mars rover. Anyway, he would attribute the success of his videos to a unique storytelling strategy.
How do you make people feel something during the journey? How do you establish the problem, the tension, to ultimately get that payoff to be even more inspiring as a result?
There's something I've heard people call like Jenga storytelling, which is like the start of my videos usually is like, I'll tease that and I'll show them like, this is what we're building up to. There's an amazing thumbnail that got you to click on it. I want to pay off on that thumbnail in the first 30 seconds. So you know I'm not clickbaiting you. But a lot of times in my videos, if you've watched a bunch, a lot of times I'll be like, but before we get to that, we first need to go back 8 months because that's when, the package was first stolen off my porch.
Yeah.
Or that's when the squirrels started eating all my bird seed. I show you this is where I landed, but you don't even understand, dude. There's a much bigger story behind this. Just go with me. And then I take them back in time and I build up to that.
So this is a really powerful storytelling strategy, I think. And Mark actually has also talked about another thing he tries to do with his videos is hiding the vegetables. Now,
I think the key to really learning something is to not frame it as learning. So on my YouTube channel, I like to do what I call hide the vegetables. So you might see a really clickbait thumbnail of a 15-ton Jello pool, and it's like, well, that's real interesting. I'm gonna click on that. This is the world's first ever actual pool of Jello. Before you know it, you're learning about the scientific method and chemistry. I have a handful of teachers that not only shaped my own learning process, but shaped the way I teach now. Give me 1 second. You asked me about my teachers. I literally get a little bit emotional. Shout out Mr. Malloy, my AP high school statistics teacher. Hands down, teachers are my favorite profession. I think they're incredibly important. At some point, I'm going to retire from all this and be like a volunteer high school physics teacher. That's my dream job.
now, if you're a parent, you know exactly what Mark is talking about. Sometimes your children don't want to eat their vegetables. And this is something that's a pretty common experience for many parents out there. So you do everything that you can to hide vegetables in foods that your children enjoy. So you're putting pumpkin or zucchini into muffins. You're sneaking broccoli into pasta sauce. You're doing everything that you can to get something healthy into your children in a way that it's presented to them that they like, right? That they're enjoying. And Mark's talking about doing that with his videos. He's sneaking science, he's sneaking STEM content into his videos. He's grabbing your attention with this opening, like, boom, here's this amazing clip of porch pirate stealing a package off of his front porch, or here are these squirrels that are raiding his bird feeders and they're taking all the bird seed. And then he's telling us, hey, we want to back up and tell the story, right? Let's go back 6 months. Let's go back 8 months. And when he tells you the story, it's not just the porch pirates. It's not just the squirrels completing an obstacle course. He's building in STEM, engineering, math, science content. So you're learning while you're watching this video that's highly engaging and entertaining. My children are huge fans of Mark Rober. In fact, during COVID, they got really hooked on his squirrel obstacle course or backyard squirrel maze videos. And they, after watching a number of those videos, said, Dad, we've got squirrels. We need to do this, right? So they talked me into building a squirrel maze in our very own backyard. And it was really cool. It was a fun experience. They learned so much. They created the designs. They learned a ton about physics and engineering. And then, when we had built it, they learned that it wasn't such an easy thing to say, here's this maze, squirrels, here are these walnuts at the end of the maze, just go do it. It doesn't work that way. They had to learn, in fact, how we train the squirrels in order to get them to progress through the different parts of the maze. So they were learning a ton as they went through that experience. And we actually shared some things that we learned online about the backyard squirrel maze. One of the things, unfortunately, that happened is as you train the squirrels to complete the obstacle course, they leave a lot of presents for you on the different platforms. So, yeah, we had to do a lot of cleaning. I think hiding the vegetables is maybe something that we can do to improve the level of engagement that we're getting from our students. So, I want to explain to you, how do you build a unit for AP Biology that's going to retain a high level of rigor, that's going to teach what's in the CED, that's going to prepare students to do well in the AP exam, but you're grabbing students with a Jenga storytelling approach and you're hiding the vegetables along the way. Why should I teach with storylines? Well, number one, they're fun and they are interesting. They are engaging and they use inquiry to answer questions. And these are questions that students are asking. And they raise engagement level and achievement. All right. Now, as I presented in NSTA, I was joined with a couple of my colleagues who I'd like to recognize here because they played a huge role in helping me develop this presentation. So I worked along with Josh Anoff, who teaches biology and AP biology at Severn Run High School in Severn, Maryland, and Dr. Angela Tatum, who is one of our science academic specialists in Anne Arundel County, where I currently work as a science teacher. When we're thinking about reaching students, we want to talk about phenomena, okay? Now, how do you reach people on YouTube? Well, you do that with thumbnails. You do that with titles, right? And Mark would probably, or any other YouTuber for that matter, would talk about the importance of titles and thumbnails to increase the clicks and the views that they're going to get on their videos. And the idea here is we want students to click on our lessons. We want students to click on our units. How do we do that? Well, I'd like you to consider the two thumbnails that you see on screen. One titled The Chemistry of Life, the other titled Survivor, American Southwest. Now, if you gave students these two options and said, these are thumbnails of Unit 1 in AP Biology, and you can choose which one to participate in, and your learning is going to be at an equally high level of rigor. One is not easier than the other. One is not teaching you less than the other. Those factors are equal. Which one do you think that students would want to click? The goal of storylining is to move away from students feeling like, I need to learn this, where the motivating factor for them is primarily external, to, I want to find answers to questions that I'm asking about a topic, because then the motivation becomes much more internal for the students. And this is when I think you'll see for the majority of students that their level of engagement will increase. How do we start this unit, right? So we're teaching Unit 1. It's titled Survivor, American Southwest. We have made our choice. We've clicked on this thumbnail. Now, the opening day of the unit, I will show a trailer video to my students, and I will ask them to view the video And then after they're done watching the video, I want them to ask thoughtful questions about predators, evolution, water, temperature, chemicals, and then macromolecules like carbohydrates and lipids, proteins and genes. The goal of this is to have students generate questions that will drive their learning throughout the unit. Let's go ahead and check out the trailer. I have the students take a few minutes to work individually to ask some questions that the video is sparking in their thinking about what they saw, right? Now, there's a lot going on in this video, and I think that it's going to open up students to ask a lot of questions about things like camouflage, things like how do they survive when it gets really hot? How are these animals able to survive when there's not a lot of water available in their environment? So the students will work independently for 2 minutes to generate a set of 3 questions. Again, thoughtful questions about the material that they've been presented in the trailer video. Then I have my students grouped, typically groups of 4, sometimes groups of 3, and I'll ask those groups to talk about their questions and to, as a group, come up with their top 3 questions that they would like to ask about the content that we'll be learning about in the unit. I have them submit using a QR code. So they scan that and they go to a Google form, and the group will submit 3 questions. Now, I've got that set up so that in my classroom, I can just very easily do a copy paste into this Google slide and populate what we call a driving question board. Let me show you an example of one that was created by my students earlier this year. And we can use those to drive the lessons, but it's also driving engagement because what we're going to do is tie the lessons to questions that students are asking. You know, is there a disadvantage to having fur? How do they get enough water? So there's many questions that the students will generate, and that's going to drive the lessons, the labs, and the activities that we do through the unit. This is an example of some reflection work that I have students do with their driving question boards. This is actually from a later unit. This is from Unit 8, Ecology, where students are learning about the wolves and moose of Isle Royale in Michigan. We'll talk more about this one a little bit later on in the presentation, so we'll come back to it. Let's talk about one of the really important aspects of storylining. It's what we call a coherent lesson sequence. Now, what we're doing in this unit, it's unit 1 of AP Biology. And my goal in writing these storyline units was to really work within the construct of AP Classroom, to work within the CED. So the units primarily are existing within a unit of the CED. So in Unit 1, Storyline Survivor, we're doing Unit 1 of the CED. We're not bouncing around from one unit to another to another. And I think that that's important for a number of reasons. Okay, so what's that coherent sequence of lessons going to look like? So the first day of the unit, we start with the driving question board, the DQB, where students are introed to the topic and they generate a number of questions about that topic. Then we move into an activity called Mouse Hunter that you're going to see here in just a little bit. In Mouse Hunter, the students are going to learn some basics about natural selection that we'll revisit later in the year during Unit 7. The students also will set up in Unit 1 a seed hydration experiment. We'll talk more about that, but they're able to work on asking questions, learning about null hypothesis, their practice writing FRQs, and then we can move into those topics of Unit 1. So we are getting the properties of water. We're learning about surface tension, evaporative cooling. There's an ability to work in a number of activities. And we're also learning about the chemistry of life. We're moving into the elements and the macromolecules that are critical for life processes. Students are learning about polarity. They're learning about hydrolysis, dehydration, synthesis. They're able to reference and use the AP Classroom resources, and they're able to do it in a way that makes a whole lot of sense to them. Because again, Unit 1, yes, we're including a tiny bit of 7 in here to give it a framing that's interesting for the students, right? If we only teach macromolecules, water, and the elements of life, I got to be honest, like that first unit is really dry. And I'm sorry, the irony there, right? It's a unit about the properties of water, but it's really dry content. It's not something that a lot of students are going to be really excited to learn about. But when we bring in that context of some natural history, these rock pocket mice and kangaroo rats, and how do they survive in this really challenging environment, I think we build a better level of engagement and buy-in for why do I want to learn about this? So we're learning about those macromolecules. Later in the unit, the students will be completing that seed hydration experiment. More about that in future slides. And they're able to use, the personal progress checks, FRQ, multiple choice from AP Classroom. And then we do some assessment. Now I have more detailed information about assessments later on. Again, I think a really important thing here, the way that these units are structured, is the storyline lives within a unit of the CED. I think that this is really important for students because then it's a logical progression. They learn about properties of water, they learn about the elements of life, they learn about the macromolecules, and they're able to fit it into the context of that natural history piece, the part of natural selection that we introduced at the beginning of the year. And then you're able to bring in evolution and natural selection throughout the remaining units until you get to 7 at the end of the year. Okay, let's keep looking here. Now, in this one, we're going to talk about the activity called Mouse Hunter. There's a classroom version of Mouse Hunter. There's also a digital online version of Mouse Hunter. So I'll show you clips from both of those here. The basic idea is that you have students collecting data to evaluate if camouflage provides a survival advantage to prey species. So our classroom model uses dots. So we have different color sheets of paper, black, white, and tan, and we spread 10, that's always hard to say for me. We spread 10 tan dots on the sheet of paper. Now, we spread them onto the white paper, and a student who is playing the role of a hunter will put on these scuffle vision goggles that we use in class that obscure their vision a little bit, and they'll wet their finger and try to quickly remove the 10 dots from the paper. The time that it takes for them to remove all those dots is the hunting time. We have the same predator repeat that process using all 3 colors of the paper field. And then we evaluate the survival time of the tan dots. Do they survive for a longer period of time on the black paper, the white paper, or the tan paper? And we can do a lot of good statistics to evaluate, is there a benefit to camouflage? Is there a benefit for these prey species when their fur color blends into the color of their surroundings? Okay, let's go ahead here and take a look at some veggies. Okay, so let's talk about the veggies. This is a sample graph of data that my students have collected over the past few years running this experiment. So it's based on 53 different students completing this. So this is the past 2 years of my students completing this experiment. And we want to think about, back to Mark, hiding vegetables, right? Hiding vegetables in his video. We're hiding vegetables in this unit, right? We're gathering, or we're getting the students to, get hooked, right? They're clicking on our unit because they want to know about these mice. They want to know how they survive in their desert, challenging environment with so many predators around. So the vegetables that are hiding in this unit, we've got science practices covered where students are posing scientific questions, they're learning methods, including writing scientific procedures and carrying out those procedures. They learn how to represent data by doing graphs, so they're learning how to create proper scaled graphs, proper graph type, how to use the different statistical tests and error bars. They're also covering big ideas here. We're learning about evolution and energetics and systems interactions throughout this unit. The learning objectives, we're really tying that into natural selection. Now, it's not a full coverage of natural selection at this point of the school year. We come back to that in Unit 7 and give it a more complete coverage with a follow-up activity that's built off of the initial mouse hunter activity. The later activity is called the rock pocket dots, really connected to the rock pocket mice of the American Southwest. So after students have completed the mouse hunter activity, there's the most important experiment in the unit where we're really going to tie together properties of water, chemicals of life, chemistry of life, and the macromolecules. So in this experiment, students are analyzing the hygroscopic properties of seeds. The basic setup for this experiment has students testing seeds in simulated conditions to mimic what is present in the environment where these mice and rodents live. So the students will get different samples of seeds. We use cumin, coriander, and pepitas, right? So it's very Southwestern themed seeds. And the students will place those seeds into two different environments. So they put approximately a 5-gram sample of seeds into a petri dish, and one petri dish will be left out in the classroom. You can see those right over here. And those are in dry conditions. This is simulating the conditions above ground. Now, the other seeds that the students will test, again, they're measuring out approximately a 5-gram sample, is to mimic what is going on in the burrows. You see, these animals are granivores. They eat seeds, but they don't eat them right away when they find them. They actually gather them up in these pockets in their cheeks, right? And they bring them underground into their burrows and they store them for a period of time before they eat them. Now, the seeds are actually going to act like the little packets when you buy a new pair of shoes. So that packet they put in the box is full of chemicals that will absorb moisture directly out of the air. The seeds will actually do this too. So when we place the seeds into the humid burrow air, the simulated burrow conditions, we're going to see those seeds actually gain mass over a window of time of approximately 1 week. And the students will leave their samples. And while this experiment is running, they're learning all about properties of life. They're learning about the elements of, key macromolecules, and then they learn about the structure and function of those different macromolecules. And then at the end of the unit, they're going to come back and look at these samples and do a comparison of the different seed types. Do some types of seeds absorb more water? Would there be a survival benefit to the rodents to select certain types of seeds as opposed to other kinds of seeds? And our question is driven around what kind of macromolecules are present in high levels in the seeds that absorb a high amount of water. We've got more veggies coming with this lab, right? So it's a lot of the same science practices, big ideas, and learning objectives that we first introduced with Mouse Hunter. So students are asking scientific questions. Again, they're learning how to represent data with graphs, including error bars. They're practicing those statistical tests, including the standard error of the mean. And there's other concepts that are covered in this experiment. So they're learning properties of water. They're learning about evaporation. They're learning about hydrogen bonding. They're learning and connecting that to the structure of biological macromolecules, including the really important ones that are found in these seeds, which are carbohydrates, lipids, and proteins. They're also going to learn how to calculate a percent change in mass. That's how they'll evaluate the amount of change in the seed samples. And this is really important because when we get to Unit 2, doing the potato or carrot osmosis experiment, your students will be better prepared for that lab because they've already done a percent change in mass calculation. Finally, they're learning the differences of hydrophobic versus hydrophilic interaction that goes along with hydrogen bonding. And we know that this is really important for students to understand as you move forward into future units like Unit 6. Along with these structures, during the unit, students are completing homework and participating in what I call left side processing. So on this page, this is a snapshot of what a student's notebook would look like for actually Unit 2. Now, Unit 1 sets up the same, but I wanted to show you an example of how these storylines are connected to each other. So in Unit 2, students are learning about cell size. So I typically will assign a reading from a textbook or to watch the daily videos, and I have students take notes in a Cornell style. I'm giving them some structure, so I'm asking them to meet some key learning goals, like explain the effect of surface area to volume ratio, asking some key questions, and giving them important vocabulary, terminology to use in their notes. So that's completed outside of class. When we are going to use those concepts in class, I'll have the students complete what I call left side processing. That's the 2.3 cell size that you see on the left of your screen with the picture of the moose and the mouse. So in Unit 2, we're going to connect back to the mice from Unit 1. We're also going to connect to the moose that are featured prominently in Unit 8 when we explore the ecology of Isle Royale, a national park in Michigan, located actually on an island in Lake Superior. So what's the connection of all of these things? Well, in Unit 2, we're really looking at the relationship of surface area to volume as it relates to the rate of diffusion of materials in or out of cells. But surface area to volume ratio is also important in the temperature regulation of different species. So there's a very big difference in temperature regulation in an animal that's small, like a mouse, versus an animal that's really big, like a moose. So we can explore those relationships in Unit 1. We don't really actually introduce it and say we're looking at this, but we're starting to kind of lay the groundwork to bring it back in Unit 2 and again in Unit 8. And we can always reference that earlier learning that way. Let's talk about ways that you can assess learning during this storyline unit. Okay, so this one here. Earlier you saw how the students are going to create what we call a driving question board. So I use the driving question boards as formative assessments as I go through the unit. So I will make print copies of the driving question board. And remember, this is generated for the whole class, right? So each group in my class is going to get a printed copy of the driving question board. And I'll typically pass those out during the class period. And we will identify questions together that we're going to answer that day in class. And I think this really drives engagement because students see that the lessons that they're participating in, the labs, the POGLs, are going to help them answer questions that they first were wondering about at the beginning of the unit. When they use these, I will have them sometimes record new questions on the margins. So a lot of times they'll generate ideas as they're learning in the unit so they can fine tune questions or they can add to questions that they've asked earlier. I'll also have them write post-it reviews, asking them to connect their learning from that day to the driving question board. What have you learned? What questions can you answer? What questions can you now partially answer? What more do you need to know to give a more full answer to some of those questions? So this is a great way for me to check in with my students to see what they're learning, to gauge their level of understanding, and to then, develop ideas for that next lesson. How do I help build their understanding even further? This is another way that we're assessing student learning with the storyline units. So this is an example of a CER, claim evidence reasoning, lab poster. This one is one example that my students made last year for the hygroscopic properties of seeds experiment. So they have a research question: what kind of seed collection behavior, would be adaptive to the rock pocket mice. Is it more beneficial for them to gather seeds that are higher in lipids? Is it more beneficial to gather seeds that are higher in carbohydrates? Or maybe, higher in proteins? So we're going to answer that question. And you can see that they're able to display their data in tables and graphs. They're interpreting patterns and forming conclusions based on statistical analysis to make a claim about what kind of seed would actually most benefit these animals who amazingly, I didn't know this a few years back, but rock pocket mice and kangaroo rats, they don't drink water. All the water that they need to maintain water homeostasis in their bodies is actually uh, from the food that they eat. They don't drink water. So this is a really good way that I can assess my students' learning through the unit, right? We can do additional things too. So in many units, we'll include spiderweb discussions, which is a type of Socratic seminar activity. And then we even have final models that students are creating. Okay, so let's go ahead and take a look at a final model here. So this one is a final model that was created for Unit 1, looking at the adaptations that these rodents have that help them survive in their hostile desert environment where there are predators and scarce water and these huge temperature swings, right? So this student submitted a final model that I highlighted one part of what they wrote, but they said that the seeds that mice store are made mostly of hydrophilic carbohydrates, which attract water. And you need to see this beyond like the lasers coming out of the raptors, the eagles' eyes. This part right here. Look at this. Okay, so we're seeing those seeds that are high in carbohydrate content really just like reaching out and grabbing that water that water vapor that's present in the burrow. These animals will actually plug the entrances to their burrows to boost the humidity level so that there's more and more water being absorbed into the seeds. Okay, so this I think says something really important about what storylines can do. When you're looking at this final model, I think what you're seeing is evidence that this is a student who learned a lot, but also had fun. And that's really important because then at the end of that unit, I think the student is left saying, what's coming up next? What do I get to learn about now? I want to send out a special thank you to Lisa England, a teacher at Washington Community High School in Washington, Illinois. Lisa was part of our team that was submitting the proposal to present at NSTA. Lisa was one of the early adopters of the storylines that I wrote. And she was going to be part of our presentation team, but then was not able to secure funding to help pay for her travel. So she wound up not presenting with us, but she played a really important role in putting together the proposal materials that did help us get our proposal accepted by NSTA. So thank you, Lisa, and also thank you to your student who was happy to share their work and have it included in our materials. Okay, now at the close of a YouTube video, from many YouTubers, you're going to hear them say something like, if you enjoyed learning about the properties of water, you might like this video too. And the goal of those YouTubers is to get their viewer who's made it to the end of their video to click on their next video, right? Because that's important for them. That's how they're generating income. Well, we want our students to click on our next unit, right? So if we can get our students to click on our follow-up unit, and in fact, this one, we get to kind of continue the story about the importance of water. I'd like to do some acknowledgments here. So the Anne Arundel County Public School System, where I currently work, Vicki Romanowski, who is the coordinator of science in my county, Lisa England at Washington Community High School in Washington, Illinois, Michael Nachman, Bridget Von Holt, John Molt, Rolf Peterson, Sarah Hoy, Paul Anderson, and Wilhelm Bohr. These are scientists who have volunteered their time to meet with me virtually to record interviews that are incorporated into the units. And you'll find some of those videos already on my YouTube channel and website. Some of them I'm still working on, and they'll be available for the upcoming school year. Finally, I'd like to acknowledge my family for their support and Peanut, our beloved family pet, who was the original inspiration for the hygroscopic seed lab and inspired me to write the Survivor American Southwest storyline. At the very end, don't forget to like, subscribe, comment, and drop a question for me down below. And you can find all kinds of places to interact with the AP Biology storylines that we're creating here at Lab Hamster. Thank you for checking out this video, and I hope to connect with you real soon.